everybody. Today in this lecture video, we are going to focus on the urinary system. Specifically, this is going to focus a bit more on the basic anatomy of the urinary system, both at the gross level, and then we'll take a little dive into the microscopic level to examine the nephron in order to prepare us for a future lecture on the physiology of the urinary system, or how we're going to actually create urine. So, big picture, most of you already know this, but never hurts to make sure we're all on the same page. We've got 4 major organs in the urinary system. We have the kidneys, the ureters, the bladder, and the urethra. So if we take a peek over here in this diagram, we can see our kidneys here with the hilum, where the renal vein is exiting and the ureters are exiting and the renal artery would be entering. We can see the ureter connecting the kidneys down to the bladder, and then the bladder is drained by the urethra. The urinary system has several major functions. I think you already know that it plays an important role in adjusting blood volume and pressure levels. It may not address the root cause of those blood volume and pressure levels, but if blood pressure is low, one way we can increase it is by increasing the volume of the blood. And the way we could do that is by not releasing as much water. Or conversely, if blood pressure is really high, we could reduce that blood pressure by lowering the volume of the blood by taking more water out of it in the kidneys and peeing that out, and thereby decreasing blood pressure. Again, it may not be addressing the root cause, but it's a quick way of adjusting blood pressure levels. We are also going to see the urinary system help regulate blood plasma concentrations, because one of the things the kidneys are going to do is either hold onto or excrete things like sodium and potassium and chloride. So what happens in the kidney with those electrolytes will determine what's going on in terms of concentrations of those electrolytes in the blood plasma. The kidneys are also good at moving hydrogen ions, so it can impact and help stabilize blood pH. The kidneys can help conserve valuable nutrients such as glucose by making sure that we retrieve any glucose that might be in the filtrate of the nephron, and returning it back to the blood supply. And then also drugs, toxins, and or their metabolites can be excreted through the kidneys. So big major functions there. All right, so let's start with the ureters. There are typically 2 of them, one from each kidney connecting down to the bladder. They are going to be lined with transitional epithelium, which we will also see in the bladder. And remember, transitional epithelium is kind of special. It's really only found in the urinary system. And you can see here, it has this interesting look to it. It's clearly stratified, but if you look along the surface, you can see there's a lot of cytoplasm on either side of these nuclei, and those are umbrella cells, and they're quite large cells, and they're capable of being very kind of poofy when the bladder is shrunk or empty, but when the bladder fills and expands, then they can stretch out much thinner to maintain that inner lining and the waterproofness of the bladder. So super important. And then just like everywhere else that we've looked at this semester, underneath the epithelium, if I just pull this over a little bit, underneath the epithelium, we are going to see that we have a lamina propria, just that loose areolar connective tissue that supports the epithelium. Together, they make up the mucosa, and then outside or to that mucosal layer, we are going to have some smooth muscle, and then outside of that, we'll see that outer connective tissue layer. So pretty typical, not too different from the digestive system. Each system has its own, particular characteristics, but the general pattern is the same. All right, that brings us to the bladder, which of course is for storing the urine that the ureters deliver from the kidneys. The bladder has rugae, just like the stomach. That is going to allow for the bladder to expand when it fills with urine. Again, it's going to have that transitional epithelium that I just spoke about, capable of expanding and getting thin, but still remaining waterproof. You'll notice that the ureters actually come in at the bottom, which is maybe not intuitive, but as a result of them being towards the bottom of the bladder, they have these unique slit-like openings instead of round openings, and that's important because it helps prevent the backflow of urine as it fills the bladder from backflowing back into the ureters and up towards the kidneys, which would put pressure on the kidneys, and we don't want that. So if you look at this image of the bladder, you can clearly see it's got its inner epithelial lining. It's going to have the muscle that surrounds it, the detrusor muscle. You can see the ureters coming in behind the bladder, again, entering down at the base. You can see there are several ligaments. You've got the middle umbilical ligament and the lateral umbilical ligaments that help hold the bladder in place. In the male, we're going to see that urethra exiting from the inferior aspect of the bladder is going to be surrounded by the prostate. We've talked about clinical issues related to that in a previous chapter. Obviously, we don't have a prostate in females. And then here in this image, we're just seeing a micrograph or a slide under the microscope of the bladder, showing again that transitional epithelium with the underlying lamina propria, a little bit of submucosa under that. You got that big layer of muscle, or the detrusor muscle is its special name. And on the outside, you have some more connective tissue, which would be the visceral peritoneum. Remember, the parietal peritoneum is what lines the peritoneal cavity. The visceral peritoneum is the other layer of that serosa that's on the actual organs. The urethra will transport urine from the bladder and out of the body. In the male, the urethra is going to pass through the prostate on the inferior aspect of the bladder and then through the penis. In the female, we're going to see it exiting the bladder, and it will exit anterior to the vaginal canal. So you can see the relationship of the bladder and the uterus, which definitely explains why when pregnant, there is often the urge to urinate because baby's weight is literally pushing down on that bladder. We got a fancy word for urination. It's called micturition. It is governed by two spinal reflexes, both of which are located in the brainstem, specifically the pons. And hopefully you remember from AP1 that the brainstem has a lot of autonomic nervous system function. So a lot of centers that are going to govern things that you don't consciously think about doing. So think about heart rate, blood pressure, and things like that, but also urination. So you have one pontine center. I really don't think it's supposed to say storage center there. I'm not sure what it's supposed to say. This is definitely an autocorrect issue going on here, but we have one pontine center that's going to focus on inhibiting urination, and we'll have the pontine micturition center, which will stimulate urination. So sympathetic will hold on to urine, and parasympathetic will release urine. And if that's counterintuitive, think about it this way. If you're running from a bear, that's sympathetic nervous system, and you'd probably rather not be wasting your time peeing your pants. I mean, you might, but you're probably hoping not to. So when you're more relaxed and have time to rest and digest, that's a good time for urination. Again, both of these centers are located in the pons, all autonomic nervous system. And I thought this would be a good time, since we're talking about micturition or urination here, to just throw in some key vocab that's associated with the clinical setting and urination. So we have polyuria, when you're peeing too much, right? Poly means much or many. Anuria, which means not peeing very much at all, less than 50 mils a day, because A means no. Incontinence means you're not able to control it, so you might have normal urine production, but you're not able to go when you want to, or hold it when you want to. And dysuria is when you have painful urination. So some important clinical terms. Your kidneys are located retroperitoneally, which hopefully you remember from previous discussions. That means they are behind the parietal perineum. peritoneum. And so if you look in these images, you can see here that the kidneys are tucked up under the 11th and 12th ribs and then extend inferior to those ribs. So they're partially protected, but this might explain why they have a very tough outer fibrous capsule made of collagen fibers. And then wrapped around that, they're going to have some perinephric fat, so a layer of fat, and then also some renal fascia. All of that is providing some protection and shock absorption since they aren't fully encased in any kind of bony encasement, so they are at risk of damage from impact. Again, just pointing out here that we have that hilum in the center where our renal arteries and veins and nerves will enter or leave, and the ureter, of course, is leaving. And typically speaking, we're going to have two kidneys, so we want to make sure that we label them right and left when we are talking about them. But it is possible to be born with a single kidney, and you can definitely live a great life with just one kidney. So if we take a look at the gross anatomy of the kidney, so imagine we just sort of slice it in half and we open it up, what would we see? Well, we'd see something like this diagram here. We'd be able to see very thin outer fibrous capsule that I mentioned just before, and then underneath that capsule, we'd see what we call the renal cortex. And then below the renal cortex, we would have the renal medulla. The renal medulla is actually made up of renal pyramids with columns of cortex in between. So these are renal columns and these are renal pyramids. All together, they give us the renal medulla. And again, the cortex is just the area of the kidney outside of the renal medulla. Half of a column, a pyramid, and half of the next column, plus the cortex above, is considered a lobe of the kidney, or a kidney lobe, of which there are typically 6 to 8 of them. So those are the major parts of the kidney. You can also see the very sort of pointy part of the renal pyramid is called the vena papilla, which dumps into the minor calyx. When two minor calyxes come together, that would give us a major calyx. And then where they all come together will be the renal pelvis, and ultimately that will drain to the ureter. So that's where the urine is going to drain out of the kidney. Here it is in real life, if you will. So you can see these renal pyramids, and you can see the renal cortex, right here, the cortex and the renal columns in between the pyramids. So from here to here, all the way around would be the medulla, and then outside of that would be the cortex, and then like right here would be a lobe. We've got a minor calyx there, and a minor calyx here, and a major calyx, and then the renal pelvis, and then the ureter leaving. And of course, that very outermost layer would be our fibrous capsule. So you can see all the same structures in this real kidney as we could in the kind of cartoon diagram on the previous slide. Now, the microscopic anatomy of the kidney is a little bit trickier. You definitely can't see it here in this kidney, and even looking at it under the microscope is challenging in terms of teaching it, because we're going to catch it in all different planes of section, making it really hard to envision what the nephron looks like. So instead, we're going to use, sort of these cartoon diagrams so you can get a sense of not just what it looks like structurally, but then also relate that structure to its function as we move into future lectures. So in terms of the microscopic anatomy of the kidney, we use the nephron, or we identify the nephron as the functional unit of the kidney. It's what is going to make or produce urine. There are two types of nephrons. We're going to have cortical nephrons, and we're going to have juxtamedullary nephrons. Watch out for the word juxtamedullary, because that's not the only place you're going to see it. There are also juxtamedullary cells in the kidney, which play an important role in detecting blood volume and blood pressure. They are located in the glomerulus, and I think we mentioned them back in the endocrine chapter, so just a flashback to that. So the cortical nephrons, which are most nephrons, about 85%, the majority of the nephron is located in the cortex. So if you look at this image right here, most of the nephron is located in the cortex, with just a little bit of this loop part located in the renal medulla. But with the juxtamedullary nephrons, only a small portion of it is located in the cortex, and the overwhelming majority of the loop is in the medulla, and that's why we call it a juxtamedullary nephron. No matter which type of nephron it is, they both drain to collecting ducts, and those collecting ducts will ultimately drain what will be urine at that point into the minor calyx. So the nephron, look at me, I'm just fixing my slides as we go. So the nephron has two main parts. We can divide it up into the renal corpuscle and the renal tubule. The renal corpuscle is the kind of ball of capillaries. I always say it looks like a meatball under the microscope. But it's the ball of capillaries where we have a very high pressure due to the afferent arterial that comes in being larger than the efferent arterial which leaves. So it creates like a bottleneck which increases pressure inside that ball of capillaries. And that high pressure is really important to forcing water and solutes out of the blood and into the capsular space through the fenestrated capillaries of the glomerulus. And that's the process of filtration, which happens only in the glomerulus or the renal corpuscle. Then the nephron becomes the renal tubule, which is a big tubule that we're going to break into different parts and name each part of it. That will receive the filtrate from the renal corpuscle, and ultimately it will modify that filtrate into actual urine, which will then be eliminated from the body. The nephrons, as I mentioned, are all going to drain into collecting ducts, and those collecting ducts will ultimately deliver the urine to the calyces so that it can reach the ureter and be stored in the bladder. So let's take a look at each part of the nephron. We'll start with the renal corpuscle, also known as the glomerular capsule or the glomerulus. And we have to watch out for these words because there are some nuanced meanings here. We have a habit of using them interchangeably, but they do actually refer to specific areas. So the entire thing, and if I can make this a little bigger, the entire thing is the renal corpuscle, right? The part that surrounds it is the glomerular capsule, and you'll notice it is simple squamous epithelium. You looked at this in ANP1 in one of the first days of your lab class as an example of simple squamous epithelium. And then in the middle, we have that ball of capillaries, which we call the glomerulus. And the capillaries inside that glomerulus are going to be fenestrated capillaries. And this is where filtration will take place. And the filtrate will be captured in the capsular space, but the blood will remain in the capillaries and exit the glomerulus through the efferent arterial and ultimately ends up in the peritubular capillary network, which I will share with you in the not too distant future. All right, so that is our renal corpuscle. Next, from the renal corpuscle, once the filtrate enters the capsular space, it will then drain into the proximal convoluted tubule. The proximal convoluted tubule is the part of the nephron that is closest to the glomerulus. That's why it's called proximal. And it's got a lot of twists and turns in it, which is why it's called convoluted. We call it the PCT for short. In the PCT, and we will talk about this in a lot more detail in another lecture, there's going to be a major reabsorption of nutrients from the filtrate. For example, we will absorb glucose in the proximal convoluted tubule so that it doesn't end up in the urine. The proximal convoluted tubule is simple cuboidal epithelium, and it has microvilli. Those cells have microvilli in order to increase the surface area for absorption. From the PCT, we're going to move into the nephron loop. The nephron loop has two parts. It has a descending thin limb or thin segment and an ascending thick limb or thick segment. They are going to play important roles in water reabsorption. And you can see the thin limb is simple squamous epithelium, whereas the thick limb is simple cuboidal. Again, we're going to get into a lot more detail on how urine is made and the role of these cells in the nephron in a future lecture. Right now, we just want to learn the basic anatomy. So in this micrograph right here, we can see some examples of the thin loop where we've got nice, simple squamous lined ducts versus these simple cuboidal lined ducts. So you can really see the difference there. There's some really nice ones, right, of simple cuboidal right there. Oh, and that's a collecting duct, so that makes sense because they're actually even larger, which is why we're getting those really nice cells. Our thick ascending limbs are right here, and again, there's some really good simple cuboidal cells right there. All right, so that is our nephron loop, which brings us to the further away part of the nephron, or the distal convoluted tubule, which means it's going to have some twists and turns in it, also known as the DCT. The DCT is also going to have simple cuboidal cells, and we are going to see that its main role is going to be adjusting the tubular fluid or the filtrate that's about to become urine by reabsorption and secretion of a number of solutes and potentially water. So there's a lot of different things that can happen in the DCT to make the urine the way that we want it based on current conditions in our bodies. So looking at that as a whole and kind of putting it all together, here is that renal corpuscle. We can see the afferent arteriole entering and starting to form the glomerulus, and those capillaries will eventually exit the glomerulus as the efferent arteriole. We will have filtration taking place here in the glomerulus, and the filtrate will be captured in the capsular space. And from there, it will drain into the proximal convoluted tubule, which is simple cuboidal epithelium with microvilli. And it will drain down into the thin descending loop of Henle is the name of this loop, or the descending thin limb, which is simple squamous epithelium. It's going to go up. When it goes up, it's the ascending thin limb. Pretty happy if you know it as the thin limb. And from the thin limb, we'll transition into the thick limb, which is all ascending, simple cuboidal, and the thick ascending limb will become the distal convoluted tubule, and eventually it will drain the now pretty much what we would call urine into the collecting duct system. So now you can see all the pieces put together. And here is our collecting duct. It is its own special structure as well. So there are a whole bunch of collecting ducts in the kidneys that will drain many nephrons. So they'll have lots of urine entering them. And eventually the collecting ducts become papillary ducts down towards, so the collecting ducts are found in the renal columns and down towards I said renal columns, and what I meant was they travel through the renal pyramids, and when they reach the papillary part of the pyramid, they become the papillary duct, and then the urine will drain into the minor calyx. And they are capable of adjusting concentrations of water and solutes as well. So there's this like one last opportunity to either, reabsorb water or reabsorb a particular solute or excrete a particular solute. So it's our last chance to adjust the urine in the collecting system. Here is an image of what the collecting duct looks like. It is also simple cuboidal epithelium, but we're going to have two special kinds of cells. We're going to have intercalated cells and principal cells. And those cells are going to play important roles in secreting and reabsorbing hydrogen and bicarbonate ions. That's the intercalated cells, so that can impact pH. And then the principal cells are going to be really effective at reabsorbing water and or secreting potassium. So two different types of cells with two different functions in the collecting ducts. By the time it becomes the papillary duct, we just have, it's almost like they're kind of between cuboidal and columnar cells, and they are really just delivering fluid. These are just very traditional ducts. There's not a whole lot going on in terms of secretion or absorption. So now if we put those all together, you can see the complete flow in urine production, starting with filtrate, and then we're going to have it form urine, and then that urine will flow out the collecting system and ultimately to the renal pelvis and the ureters. So I mentioned a couple of times now that afferent and efferent arterioles that form the glomerulus. So where are those coming from? Well, our renal artery is going to enter the kidney at the hilum, and it will divide into segmental arteries. So you can see these segmental arteries here. And then each of those segmental arteries will divide into interlobar arteries that will travel up the renal columns, and then they will arc over the top of the pyramids. When they arc, they are called arcuate arteries. Those will then give rise to cortical radiate arteries, which you can see right here. And hanging off of them, it looks like little berries, are actually the glomeruli of the renal corpuscle. And so those are the afferent arterioles. And so now we can see the afferent arteriole coming in and forming the glomerulus. And then it leaves as the efferent arteriole, which is a little narrower than the afferent. So that helps build pressure inside the glomerulus. And that efferent arteriole will go out, and then it splits to form the peritubular capillary network, which surrounds the whole nephron. And ultimately, they come together to form a cortical radiate vein, and these veins will ultimately drain back to the renal vein and go back to the inferior vena cava and back to the heart. So it's a, big circuit here, but these peritubular capillaries are going to be really important as we form urine in the nephron. If we want to get rid of stuff, we can pull it out of the peritubular capillary and secrete it into the nephron tubule. Or if there's stuff in the nephron tubules that we want to retain, we can reabsorb it, move it into the peritubular capillaries, and then send it back to the blood supply via the renal vein. So it's an important network here, and so you need to know the basics of the arteries that make it up, the vessels that make it up. This is just a great overview. It's everything I've already talked about. So this is a slide that you might want to pause on and kind of read what's happening at each of these, because it's a great overview. You could also print this out, and in all the white spaces, you could add notes based on your next lecture on the physiology, which is going to get into the detail of what's in each of these boxes. So you can see in the renal corpuscle, we want to know that's where filtrate is produced. In the PCT, we're going to see the reabsorption of water, ions, and organic nutrients like glucose. In the nephron loop, also known as the loop of Henle, we're going to see reabsorption of water. We may have reabsorption of sodium and chloride ions. And then in the DCT, we're going to see secretion of ions and acids and drugs and toxins. We may have some reabsorption of some things, and ultimately all of that urine ends up in the collecting duct, where again we could have reabsorption of water or reabsorption or secretion of things like sodium and potassium and bicarbonate, and then ultimately that urine will be delivered to the minor calyx. So there's a lot of really good information here. This slide includes so much. If you added in all this white space, like the type of epithelium and the type of cells, all the stuff from the previous slides, that'd be a great way to study. I'd love it. All right, so last little bit here to set us up for physiology is thinking and reminding ourselves that the urinary system maintains homeostasis in really important ways, right? So we know it regulates the volume and composition of blood. And to do that, it typically concentrates the urine to somewhere between 855 and 1,355 milliosmolarity. I just lost the ability to speak, guys. Woo! The solute concentration per liter ends up between 855 and 1,355, typically speaking. And we're going to get rid of things in urine like urea. Remember, we make urea when we break down protein. Creatinine creatinine, which is from muscle function, and uric acid from breaking down nucleic acids. And so if you look over here in these little charts, there's some interesting things here. So for example, if you look at sodium in our blood, it's usually between 135 and 145. In the urine, it could be as low as 40 or as high as 220. That's because the urine, right, if our sodium in our blood is riding at 135, then we're not going to want to get rid of a lot of it. So our urine concentration will be really low. But let's say it was at 200 in our blood. Then in our urine, we're going to want to get that number a lot higher because we're trying to bring this number down. So that's why there's a big range here. And you're going to see the same thing in other places as well. But if we look at potassium, it's very low in our blood and pretty high in our urine because we don't want potassium to be very high in our blood. So in order to do that, we've got to increase the concentration in the urine to keep the blood potassium levels relatively low. If you look down here at these solutes like glucose, you're going to see that they are typically in our blood somewhere between 70 and 110. Sometimes it's reported as between 60 and 100 if it's like fasting measure. But you can see in our urine, it's basically zero, right? 0.009 is essentially zero. So that's why we say we shouldn't find glucose when we test for it in the blood. So when we do urinalysis with like the dipstick test in lab, your glucose test should come back zero unless someone has maybe diabetes, in which case they're not reabsorbing all of that glucose, so the urine then tests positive for glucose. We also shouldn't see lipids or any proteins in our urine, but we will see them in the blood. Over here, you can see these are all nitrogenous wastes from breaking down things that have nitrogen in them. So proteins produce, when you break down proteins, you produce ammonia, which has nitrogen in it, and that will lead to urea formation. We take the ammonia, which is kind of toxic, and we convert it to urea. So you can see we have very high concentration of urea in the urine, and we want to keep it pretty low in the blood. Same with creatinine, which is a waste product from muscle function. Uric acid, same, high in the urine, low in the blood, again from breaking down nucleic acids. And again, we don't want ammonia in our blood. Ammonia is not good for cells. It's cytotoxic, so we want to concentrate anything we can get in the blood. We want to move that ammonia into the urine and get rid of it. So that's what these charts are showing you. And to set us up for our next lecture on renal physiology, I want to leave you with these 3 processes that are going to be required for urine formation, which I've already mentioned, but I'm just going to summarize here to prep us for the next one. So in order to make urine, we are going to have 3 major processes. The first one is going to be filtration, and that's going to happen in the glomerulus. So we're going to use increased blood pressure in the glomerulus to force water and solutes across the membranes of those fenestrated capillaries in the glomerulus to capture that filtrate in the capsular space so that it can then drain into the PCT. So the second process is reabsorption. This is where we are going to take back a whole bunch of the filtrate and put it back in the blood, because a whole bunch of things that we filtered out, we don't actually want to lose them. We actually want to keep them. So there's water in the filtrate we want to hold on to. There are solutes like glucose, maybe sodium, maybe potassium that we want to hold on to. And so we're going to do that reabsorption for all the things that we want. And when we reabsorb them from the tube of the nephron or the duct of the nephron, then we'll move them back into the peritubular capillaries to return to the main blood supply. And then the third process is that there are also going to be things that we didn't filter out, but we want to get rid of. And so we're going to use secretion as a way to do that. So we can take things out of the blood of the peritubular capillary network, and we can move them across the cells that line the nephron, and then those cells can secrete those solutes into the ducts or the tubes of the nephron so we can get rid of them. And this picture right here is the right one from your book, showing the right direction. So here's the peritubular fluid. That's just the fluid around the peritubular capillary network. So there were things that we wanted to get rid of, this green solute. So transport proteins of the simple cuboidal cells of the nephron will move that solute into the cell and then into the tube or duct of the nephron so that we can excrete it. And that is it for today. And then in your physiology lecture, we'll take a little bit deeper dive into how we use these three processes: filtration, absorption, and secretion, taking advantage of the anatomy we just talked about to actually form urine.